Hello.
Hello everyone.
Hi, how are you? Oh, Susan still can't hear you.
Yeah, I'm good.
Oh, okay, can you hear me now?
Yeah, okay.
Okay, yeah.
Good. All right, so how are people doing?
Spring cleaning.
Yeah, okay. All right.
I tried the Gemini's programming,
Mm-hmm.
and it worked to give me an image of the structure, or at least part of it, but it didn't. It seemed to have mixed up on MATLAB the like columns, and the rows are flipped.
Oh, yeah.
In that, yeah. So I think that's the mistake it made. So I flipped them back around and I got another error. So I assumed that it had them flipped.
Yeah.
So I was trying it out.
Well, that's interesting. Yeah, I think yeah, playing with the code is always a challenge, right? Figuring out how exactly it works.
Yeah. Oh well. So that's part of what I've been doing, and trying to get ANSYS. I get to fight with that this week.
Oh, okay.
But I got a program called Rhino.
What's that?
They're going to let me use it. People use that to Rhino, and then the extension called Grasshopper. They use that to make tensegrities.
Oh, okay.
Yeah.
Any reason for the animal names?
It's Australian.
Oh, okay.
Yeah.
Oh, that's good. So yeah, I look forward to seeing what you can do with that.
Yeah, I'm not sure why they have names, but like that.
Yeah, that's fine. All right.
Yeah.
Well, thanks for the update. Barshan, do you have an update?
Well, I was actually writing a community post for how my community bonding period went.
Yeah.
I didn't deploy it yet, so I'll probably deploy it tonight. I'll send it in the Slack channel.
Okay, that sounds good. Yeah. Great. Yeah, Barshan presented on his.
I guess the coding period starts from tomorrow.
Yeah. Barshan presented on his community period activities and sort of the path forward. So yeah, starting tomorrow, it will start coding period. Yeah, I look forward to seeing regular updates, and so we'll communicate through our 2 meetings, and then of course we have the Slack. So that should be more than enough to get things debugged or whatever we need. All right, so let's get started here. So I wanted to talk about a few papers today. This one that I'm going to present first is sort of a classic, as they say. So this is a paper from Physica A, which is a physics journal in 2005. This is called A Cell-centric Approach to Developmental Biology, and it's by Roland Merks and James Glazier. So this is the James Glazier of CompuCell 3D fame. So this is one of the.
I've met him online. He's a character.
Yeah, he is. So this is kind of thinking about this cellular POTS model of development and how that works and what sort of the vision is for that. So let's go through the abstract and then see what they have to say in the paper. We talked about, a long time ago, we talked about sort of the roots of CompuCell 3D. There were a few papers in the early 90s where they started talking about cellular POTS models and their potential for modeling things like biological cells and those kinds of systems. And so that kind of evolved into CompuCell 3D and the platform. And so this talks about the sort of way to approach developmental biology, because it didn't really start with developmental biology. CompuCell 3D is much broader than that. It models any kind of cellular system or some sort of multi-cell system. So that's why they and of course their applications of development, especially embryonic development, were somewhat limited. But this is sort of the vision paper for that. So the abstract reads, explaining embryonic development of multicellular organisms requires insight into complex interactions between genetic regulation and physical generic mechanisms at multiple scales. As more physicists move into developmental biology, we need to be aware of the cultural differences between the two fields. So this is, of course, a common theme in interdisciplinary work where you have multidisciplinary groups from different fields and they have to constantly explain things to each other or explain their worldview. So, this is nothing new for us, but at the, especially as we go back in time, even today, but especially as we go back in time, this was a major theme of that kind of work where you had to stop and you had to address this sort of mismatch in expectations. So this is before people really were trained in multiple fields. So now you have programs where you have sort of physical biology programs where people learn about biology, they learn about physics, and there's really, the assumptions are aligned. So, so looking at the cultural differences between physics and developmental biology, whose concepts of explanations and models traditionally differ. This is interesting. Biologists usually, at least if you're like a hardcore developmental or hardcore molecular biologist, you're looking to identify genetic pathways and expression patterns and using that as sort of a model, a conceptual model, perhaps for further experiments. And then physicists tend to look for generic underlying principles. So physicists will look for things like power laws or summary statistics or symmetry breaking or things like that. And of course, you can add in computational scientists to this. So computational scientists are looking at things like computational structure or other types of things like that. So there's this difference between fields. It's very subtle, but sometimes can lead to misunderstandings if not addressed right away. So this is when we write papers for different audiences. So if you want to write, say, a paper on hypergraphs for a biological audience, you wouldn't want to start by dumping a bunch of technical jargon on their heads about computational structures, hypergraphs, and the mathematics. You want to give them a sort of a lead-in. And, this is something that it's hard. I had to learn how to write papers for these different audiences myself. It's just kind of like you have to kind of get them interested and then you can kind of give details, but you have to kind of walk them through it. It's not as simple as just telling people what you did like you would do in a traditional paper. Well, anyways, this has been an ongoing problem in this type of work for a long time. So that's kind of the area that they're staking out. Here we discuss how we can combine such biological and physical approaches into a cell-centered approach to developmental biology. Genetic information can only indirectly influence the morphology and physiology of multicellular organisms. DNA translates into proteins and regulatory RNA sequences which steer the biophysical properties of cells, their response to signals from neighboring cells, and the production and properties of extracellular matrix. We argue that in many aspects of biological development, cells' inner workings are irrelevant. What matters are the cell's biophysical properties, the signals it emits, and the responses to extracellular signals. So that's an interesting statement, not least of which because we had a presentation earlier this year on a model of the cell. And when you build a model of the cell, it's like, what do you want to stress as a model of the cell? Do you want to stress the molecular aspects of the cell? Do you want to express, or do you want to focus on the gene expression and the protein synthesis? Do you want to focus on the effects of that? Do you want to focus, say, on macromolecules? Do you want to focus on tissues? There are a lot of levels that you can focus on. They take the approach here that when they're interested in things that are like, the structure of the cell, and maybe the ultimate goal of a physicist, which is to extract some of these broader principles, then a lot of the inner workings of the cell are irrelevant. In other words, yeah, you can have a process that transcribes genes. You can have a process that translates those RNAs into amino acids and then synthesizes proteins. But all of those things are just kind of dials. They're parameters. And so the parameters can be tuned, and that's about all we need to worry about. We don't need to worry about the details behind it. We just need to worry about, say, how much of substance X is being produced or how much of protein X is present in the cell. And then those things matter more to the biophysical properties and the things that physicists care about than the molecular biology. I mean, some biophysicists are actually interested in the molecular biology. They're interested in things like transcription rate. They're interested in things like binding rates and things like that. But that's not what they're doing here. That's certainly not the program of the cellular POTS model. So again, their argument is what matters are the cell's biophysical properties, the signals it emits, and the responses to extracellular signals. So it's interesting that there are a number of approaches to modeling the cell that don't think this way, actually. And actually, these projects involve physicists as well as molecular biologists. I'm thinking of the attempts to model bacterial cells from their genomes up to sort of the contents of the cell and to model those in 3 dimensions. So, these we talked about the work that's been going on at the Craig Venter Institute and the University of Illinois, where they've been working on these models of the cell. And they can build these minimal cells, and they can model these minimal cells. And actually, to them, a lot of the molecular details are not irrelevant. But their aims are different. They want to produce a 3-dimensional model of a single cell and then vary that single cell appropriately. In this case, what they're trying to do is they're trying to model cells as sort of these parts of a larger system. And so that's the difference. So thus we can separate questions about genetic regulations from questions about development. First, we ask what effects a gene network has on cell phenomenology and how it operates. We then ask through which mechanisms such single-cell phenomenology directs multicellular morphogenesis and physiology. So this is their ultimate goal here. And this is the thing from which they're going to extract their higher level order rules, the multicellular morphogenesis and physiology. This approach treats the cell as the fundamental module of development. So the cell is this fundamental unit. And if you're interested in transcription, then your unit is much lower. It's probably like either genes or the regulatory machinery of the genome, or it could be, things like your polymerase or other types of mechanisms that are even more fine-grained. We discuss how the cell-centered approach, which requires significant input from computational biophysics, can assist and supplement experimental research and developmental biology. We review cell-centered approaches, focusing in particular on the cellular POTS model, and present the tissue simulation toolkit, which implements the CPM. Okay, so this is just kind of an interesting aside about how fields are named. So again, we're talking about the distinction between biological physics and biophysics. And so it says here, as physicists attempt to apply physical approaches to elucidating biological problems. So biophysics is the study of physical properties of biology, whereas biological physics is where you're actually trying to answer biological problems using physics. It's a subtle difference, but it's an important distinction because the worldviews are different. So if we're doing biological physics instead of biophysics, we need to be sensitive to the fundamental differences between biological and physical epistemology, which reflects the different histories of the two fields. So they focus in on the term model, which can mean two different things. And so in biology, there's an experimental model where you have model organisms that exhibit certain phenomena that you might be interested in studying. So one of the reasons why we use C. elegans as a model organism is because, not because we're interested, well, some people are interested in nematode biology, but other people are interested in those broader concepts. And the analogies to humans in a system that's much easier to control and breed in a lab. And so that's the case for most of our model organisms. They make things easier to understand in some way. So this stands in contrast to a theoretical model, or simply a model which is a conceptual model, which describes a hypothetical mechanism using text and qualitative diagrams of interactions. So he leaves out theoretical biology here, which is an interesting offshoot of theoretical or biological physics, because it's basically asking biological questions from a theoretical perspective. But anyways, such a model is successful, and they're talking about the latter, the theoretical model, the physical, quote-unquote physical model, if it is compatible with qualitative observations and suggests further experiments that could support or reject its hypotheses. So this is where we have models that predict certain things about a phenomenon. You build a model, it's very general, and then you apply it to specific cases. So I could build a theoretical model of organisms that say that morphogenesis happens in a certain way. And in fact, our theories of morphogenesis are very general like that. They can be transported from organism to organism, and they basically describe pattern formation. And so that's kind of an example of a theoretical model in biology. It's very different from the sort of experimental model that we might use to sort of reveal or examine morphogenesis in specific organisms. So that's kind of what we're dealing with here. And then, of course, you can apply your theoretical model to different cases, and you can support or reject those hypotheses. And then you can, make a note of how many times your theoretical model were supported by the data, and you can then declare that it's either a success or a failure. So that's kind of where they're coming from here in terms of epistemology. Here they have a figure showing the systems biology approach, which, and again, this is at a time when systems biology was sort of in its formative years, where we were getting a lot of genome data, we were getting a lot of high throughput data, and we could actually measure some of the things that people had theories about. So it was an interesting time in 2005 in biology and experimental biology, and we're getting the systems biology field that's emerging. So they're proposing this model for understanding from a systems biology approach, which is essentially this theoretical biology approach, injecting physics into the mix, and in fact, also injecting computation into the mix because we're dealing with very large data sets. So into this model, we have oversimplified models, disconnected experimental facts, which is, I think, the case in a lot of biology, where you might have a very general model, but it's very simple, and it doesn't really have a lot of detail. So it's hard to make it a predictive in any sense because it just doesn't have the details. You do have a lot of details in biology, but they're all disconnected, and they're hard to sort of fashion into a complex model, something complex enough to be predictive. And so then the challenge here is to take sort of this some of these data or take some of the facts and shape them into some sort of predictive theory. So that means that we have the identification of key molecular and cellular players, the measurement of cellular phenomenology. That leads us to integrating this into a computational model, which could be this cellular plots model or something else. Then you have validation of patterns and statistical observables that feeds back to identification and measurement. So you're validating the things that you're identifying and measuring, and you're sort of, nailing down that detail of the model as something that's going to be predictive. Then you can do things like high throughput parameter searches and suggestions of further experimental work to make this more, more usable to people in the field. And then that feeds back, of course, our identification and measurement because it refines it further. So what you get out of this integration process is an integrated understanding of mechanisms of development. So they're really kind of getting at sort of the way to set up these cellular POTS models. You don't just simply pull it out and start using it and looking at the patterns. You have to do a lot of work in advance of that to try to define your problem, to try to define your model. And so paradoxically then, additional information has been made the most central biological tool for generating new knowledge and understanding. And this is called the conceptual model. And giving more information about this has made these kind of conceptual models much less helpful. So to correctly integrate this wealth of information and to use it to generate new hypotheses, systems biology extends traditional conceptual models into mathematical models. So our conceptual models are useful when they're very broad. But when you get more information, you need to figure out how to make that more predictive. Otherwise, it's just a huge collection of facts. It doesn't really give you any sort of way to use it. So we have to turn these conceptual models into mathematical models that make predictions that state formally what the relationships between the informational facts are. Because such models are generally too complex for analytic solutions, we translate them into sets of algorithms or computational models, which we then implement as simulations. And this is where we're getting to the cellular POTS model and the stuff that they're doing with James Glazier's group at Indiana. These problems are particularly evident when we focus our attention on developmental biology, where a genetic essentialism called positional information, which the intellectual school founded by Lewis Wolpert has pioneered and championed, has come to dominate. However useful, the concept of positional information has led to an unfortunate focus on genetic experiment at the expense of other types of measurement. And because this is positional information is sort of an way to operationalize this sort of concept of, how does an embryo know what position in space, how do cells know their relative position to other cells in the spatial frame that an embryo exists in. So this is something that, of course, the hypothesis is that it's some sort of thing that's pre-programmed into the genetics of the organism, that you have these different Hough genes that are active, and that's how you get these, that's how they know where their position is in the embryo. So people have focused on doing experiments to look at these genes and their expression patterns and the gradients, and then that's how they've determined positional information. But that's just
Okay,
kind
and I'm sorry.
Oh, go ahead.
They forgot the mechanical environment.
Yeah.
As well as there's a chemical and mechanical environment.
Yeah, that's what I was going to
You
get
forgot
at.
that.
Yeah. So yeah,
it's too early. Now they're including it later papers.
Oh, they don't talk about it here. Well, in their modeling, yes, they don't talk about it quite this early. You're right.
Yeah, it has to be a 2020 paper.
Okay.
Before they're starting to include the mechanics. And anyways, and I talked to him about this, and there was someone developing another model alongside his model that actually measured the forces
Okay.
involved.
Yeah.
So there were forces
Yeah.
involved, and you can't measure forces that well with his cellular POTS model,
Okay.
but you can't. Someone else was developing a model from, I think, the same institution that measured forces. So I was interested in it, but I had to do my own model,
Right. Yeah.
of course.
Yeah. So this is again, yeah. So this is like talking specifically about the Wolpert model, where they've just spent a lot of time measuring like transcription rates and other things, and so this is just kind of limiting yourself to a specific part of the explanation, right? So it turns out that, of course, when we go back to Turing's model of chemical morphogenesis back in 1952, which is one of these more conceptual models, although he does offer some route to computation, development must self-organize under the control of genetic regulation. So the genetic regulation controls things, but cells do not merely sit at a given location and respond passively to external signals. They often move over long distances and actively create the signals to which they respond. So cells are not just expressing genes or sitting in a gradient. They're moving around. And of course, as Susan said, they have mechanics that do that. So cells don't move kind of, you have to account for that movement explicitly in the model, and then by extension, the mechanics in that model. And then thus the concept of a pre-pattern in which cells migrate towards or respond to a pattern formed by an independent initiating mechanism has only limited utility. So that, one of the things that they did with a lot of the positional information work is they came up with kind of this middle ground where, okay, there's a pre-pattern that it gets involved in this. And then of course that's not as predictive as, say, like having a model where you have maybe cells migrating and actively creating their own conditions. So it depends on how you have a nice mechanism and you build a model from that. It doesn't mean that it explains the entire phenomenon. It's just something that you may be able to incorporate into a larger model, I guess is their point. It kind of reminds me of some of the attempts to do computational work with pattern formation, where you have, you build these patterns in a computer, and you say, well, how does the pattern originate? And then they come up with these templates that they will lay down, and or grids, and it kind of reinforces the pattern, but it does it in a way that's not really biological. It's just kind of like a stopgap measure they have to build into the model to get it to work. And it's like, well, that's not really explaining anything. That's just kind of like getting something to work well. So that's kind of something that we have to watch out for when we build models. Salazar Ciudad et al. Recently called such pre-pattern dependent mechanisms morphostatic, as opposed to morphodynamic mechanisms, in which cell signaling, migration, and growth occur simultaneously, and argued that morphodynamic mechanisms of evolutionary significance because they enable more phenotypic variation for less genotypic variation. Thus, to understand the dynamics of development and its evolution, we need to return to cell biology and physiology, which, of course, at the time was not in fashion because everyone was talking about genomes and their potential. So this is, again, sometimes you're doing work in an area, and there are things that are fashionable and things that are less fashionable. And so sometimes you go for the fashionable thing for funding or whatever, but that fashionable thing doesn't explain the phenomena that you're interested in. So it's kind of an interesting thing. They talk about several modeling issues here, and they talk about multi-scale modeling. And this is again in 2005. where multi-scale modeling on computers was just kind of beginning to take off. So there's a lot of more recent work on this. But basically, they sum it up as the need to characterize all the different scales of phenomenology in biology. So biological phenomena range from those which require quantum mechanical treatments at the atomic level, which is 10 to the negative 10th meters. The effect of photons on chlorophyll or rhodopsin, that's 10 to the negative 9th meters. And the classical treatment of macromolecules, which is 10 to the negative 8 meters. And then agent-based models of populations, which is 10 to the positive 3 meters. So you can see there's these very large length scales that we're dealing with. And of course, if we're dealing with a single model of space, it makes it very hard to incorporate all of this. So you need to have, say, for example, a nested model, especially if you're doing something like a cellular POTS model or a cellular automaton. You need to have a nested model to account for these different things. Because as you can imagine, if you had a cell in a model, that cell, within that cell, there's a lot of variation. And the way people usually treat that is to average out everything. And in averaging out everything, sometimes you smear a lot of the variation that makes for sort of maybe a mechanism that you're missing. So, this is a problem that you might have in some of these models where, depending on your scale of interest, you might be missing things either above you or below you. And so, again, this difference between physics and biology rears its head in that physicists are trained to believe that even very complex phenomena result from the interactions of very few, very simple underlying rules, where the complexity often results from the initial or boundary conditions. So again, this is a view of sort of biology that biologists do not share. A biologist's caricature of a physicist's approach would be to say that the physicist asserts that development is simply a problem in pattern formation, writes down a set of differential equations and a model of the fertilized egg, and tries to deduce a chicken. Or to put it another way, it's like that classic joke about the physicist who wants to explain how milk is made, and then their answer is, first we have to assume a spherical cow, which is to say that we have to kind of take a cow under ideal conditions. And of course, it doesn't necessarily explain how milk is made. It just explains sort of a homogenized process, no pun intended. So this is all kind of, this distinction between a biologist and a physicist and their sort of worldview on how problems should be stated. Okay, so he goes through more of this, talks about model validation and Occam's razor and things like that. And so it's very reflective in a lot of ways. And so this kind of gets in, this section here kind of gets into some of the programming aspects of this, because, again, we're implementing these models on a computer. So this is kind of a nice, interesting section here. Is ignoring, to a first approximation, intracellular behaviors legitimate? In a recent book review, Meinhardt states that the role of the cell as a module of development can hardly be overestimated. Indeed, much of the biology of multicellular development treats the individual cell's internal properties as a black box. So, for example, the function of an inductive signal does not depend on what happens behind the scenes. So, for example, on the number of elements in a signal transduction cascade, as long as the cells respond correctly to the signal. And then Van Dassel and Meer, in this paper from, I guess, around 2005, drew a parallel between object-oriented computer programming and modularity in biology. This is something that is oftentimes computer scientists will do to, and for the same reason physicists do it, because it's how they understand how to solve a problem, and they kind of plug in their sort of constructs and ways of doing things to the biological data. Is it well suited to it? Well, we'll see. So in this case, they're looking at the parallel between object-oriented computer programming and modularity in biology, or how biological systems often are composed of parts that are separate and interact with each other. So in biology, you'll have several modules, or an organism will be composed of several modules which interact, but also function autonomously. So they state that object-oriented programs consist of modules, or the objects. These objects have well-defined interfaces through which we address all functionality of that object. In object-oriented programs, data hiding is essential. As long as we do not alter the object's response to requests from other objects, we are free to change the object's inner workings. Mondal and Mir argue that nature cannot do data hiding, although they concluded that the closest thing to data hiding is cellular compartmentalization. So they're finding these sort of parallels between a programming language structure or a programming paradigm and biological structure. This is going to be important, of course, when you try to map biology to some sort of computational representation, because ultimately you need to implement it in code or in some form of code. And if you can make the case that a lot of the parts of our biology or our biological understanding map to these structures, then it makes it easier to find that level of representation that's ideal. So that's what they're kind of talking about here. So, then they kind of consider that, can we ignore cells and model at the level of tissues? And so their conclusion is ignoring cells is dangerous. Just one sentence. Macroscopic models, which treat tissues as continuous substances with bulk mechanical properties, and here they quote something called the physiome, which was it's an approach to looking at physiology as this self-contained thing that's sort of the level of analysis that sort of subsumes cells into these larger processes. Reproduce many biological phenomena, but fail when structure develops or functions at the cell scale. Although continuum models are computationally efficient for describing non-cellular materials like bone, extracellular matrix, fluids, and diffusing chemicals, many cell-centered models reproduce experimental observations missing from continuum models. So this is kind of their case for their approach.
Okay, so that comes to tensegrities.
Yeah.
Because the continuum models don't cover a broad enough range of elasticity to properly model living tissue.
Right. So that's their case for CompuCell 3D, I guess.
Yeah, well, it was an interesting course, and it was easy to take, and it lasted a week, and I don't know, it was interesting. I could take it again, but I think I should do ANSYS or something.
Right.
Just to try to further my thesis project a bit and get something published. So, yeah.
So yeah, then they just kind of go through this CompuCell 3D approach and how they structure this. So you're going through these different levels of biology, from the genome to the cell to the organism. The cell is the focal level. This is represented in the cellular POTS model with partial differential equations representing advection and diffusion. Then that gives you these emergent tissue patterns and morphogenesis. Then this gives you model outputs, which are predictions, possible mechanisms, or an integrated understanding of the things below the cell, beneath the cell, like gene function. The model inputs, of course, are cell behaviors, genetic regulations, perturbations to genetic regulation, and then other results of single cell modeling. So doing all of that, you can get this model of the organism, and you approach it at the right level. And of course, you have this computational framework that is suitable to the biological structure that you're dealing with.
Yeah, the newer POTS model has chemistry included as a section.
Oh yeah, so yeah, they're probably adding
They've
in
been working at it, so it's more interesting.
right. I mean, I think what they're describing in this paper is sort of the basis, or sort of the fundamental focus of
Yeah.
what they're doing. So like, they're making the argument that the cellular level is the ultimate level we need to worry about for modeling, and then we need to find something that's computationally tractable, physically interesting, because then you can get these higher level rules and other things from that.
Yeah, it will do cell sorting, for instance.
Yeah.
Surface tension sorting.
Yeah.
And I asked if they could turn off gravity, and they didn't.
They didn't think
know
that
what
was
I
a good
was
question.
talking about.
Yeah.
No, they haven't done that yet.
Why would we do that?
That
Well,
changes chemistry.
yeah.
Yeah.
All right. Okay. Did you have a question, Barshan?
Yeah, so at the beginning of the paper, you were talking about how they write the paper
Yeah.
for different kind of audiences. So does that mean like for different journals or different subjects, like biological papers, papers based on?
Oftentimes it's both. I mean, like sometimes you'll write a paper for a journal and it's like the audience is mostly like biology, right? Like you might have a journal of cell biology. That's going to be mostly biologists. And a lot of times they'll just reject a paper if it's too computational or too physical. And that's just the way it is. The readership wouldn't find it interesting or understand it. Then you have, more, I guess, interdisciplinary journals, like you have PLOS Computational Biology, or sometimes like Cell, where you write it differently for that audience. So it's more general, but something like Cell, it's going to be more focused on the biology. And a lot of times with those kind of journals, they want something for a more general audience. So they don't want a lot of deep technical explanations. Oftentimes they'll tell you to put that into a set of like supplemental materials, and they want like a more general explanation of what's going on. So it's always really hard. It's a hard dance to engage in because you never know what your audience is going to look like. If you go to different journals, the audience is different, and then if you're trying to get people to read the paper, which is ultimately your goal, you want to make it accessible to those people you think are going to be your key audiences. So if I'm like writing, and I don't want to necessarily dumb things down, that's the other challenge, is you want to make it like something that people can learn from, but you also don't want to overwhelm them with technical terms. But the bigger problem there is the worldview, right? So if you're doing like computational work or physical work, you're making certain assumptions about how things should be done, and a biologist might disagree with that approach, or it might not yield the kinds of information they want. So that's kind of a challenge, and sometimes you can't please everyone.
So basically, we have to find the perfect spot between making it very computationally detailed and also like a storytelling.
Yeah, there's a storytelling element of it, which is just kind of like because people can say, "What did I do?" and you can make a list of things, and then you'd be done. You can just make a list, but there's kind of a process that you want to go through with people and say, "This is why we did these things. This is why they're important," and then making that coherent. And then, of course, again, it's like the things that are interesting to the audience. This problem is interesting. This is maybe not so. It's trivial, and you always see that the people say it's a trivial problem, and I don't know what that means. Sometimes it means that they're not interested in it. Sometimes it means that it's not. It's kind of like I found that there are 3 million different variants of a protein. Well, that's that's okay. That's it. Not it's trivial, right? So,
My least favorite saying is, "It is obvious that,"
oh, right.
because it usually isn't obvious to me.
Yeah, that's the other thing. It's like obvious. Well, maybe not.
Not to me.
But yes, I mean, there's a lot of that's a skill that like I don't think they really teach that much. And I mean,
Yeah, it's not.
yeah, you could probably teach a class on it though, because it's like very important to get it right when you're writing
Yeah,
paper. And
presenting and storytelling is very important.
yeah, and like this is especially useful though when you're dealing with reviewers of papers. So they'll ask you about things, and they'll expect certain things, and sometimes they have bad expectations or different expectations than you, and it's like that's when you really can get some bad reviews, unfortunately.
Okay.
But anyways, yeah, that's a good question. Thanks for asking. Okay, so I think we'll go to this paper. So this paper is top-down perspectives on cell membrane potential and protein transcription. And this is a paper by Javier Cervera, Michael Levin, and Salvador Maffei. This is in Scientific Reports. This is a new paper on thinking about these levels of analysis and thinking about different problems in biology. So this one is on cell membrane potential and protein transcription, but approaching it from more of a top-down perspective or at that higher level of analysis. So we could spend a lot of time, and people do, thinking about the molecular biology of this. And the question is that strictly molecular biological approach useful to the problem, or can we get a new perspective by going up a few levels of analysis and looking at the problem? We're looking at how these things are controlled maybe by top-down mechanisms. So the abstract reads, we have explored a simple model for the multicellular interplay between bioelectricity and protein transcription using a top-down perspective that offers new insights complementary to the commonly used bottom-up descriptions. So again, a lot of ways in which we understand cell membrane potential and protein transcription are through these bottom-up descriptions. It's like, okay, you have a synthesis of different molecules, and those molecules interact, and we're very detailed in our descriptions of that, and it is obvious that that is the mechanism. But that's, of course, not obvious, nor is it even really maybe the right answer. We modeled the non-excitable cell bioelectrical representation of the external environment, including the neighboring cells, using voltage-gated ion channels and intracellular junctions. So they have a cellular model, but it's very different from what they were doing with CompuCell 3D in their cellular POTS model. They're actually modeling cells as, or populations of cells, as these ion channels and intracellular junctions, and then building a network out of that. The simulations make 3 predictions. Okay, so they're simulating this with their cell representations or cellular representations, and from these simulations they make 3 predictions. The first is that shifts in membrane potential, which you can model if you model this dynamically instead of statically. Shifts in membrane potential allow transitions between gene expression states. So we get these sort of native Markov states, basically, that we can extract from the biological data. Number 2 is in the case of different cell potential gated transcriptions, depolarized cells cannot control the distinct gene expressions as effectively as polarized cells. So we can see a difference in how the state of the cell itself controls how this process works. And we can only do that through a model of the cell and of neighboring cells because that controls the state of the cell. And 3, community effects should permit to extend the single cell control to the multicellular level. So again, community effects are the cells surrounding the focal cell that you're interested in. And thinking about that population of cells as a network where cells are affecting their neighbor cells, and so forth. This allows us to have this single cell control mechanism that transcends single cell constituents. Put it another way, if you use a model of molecular biology and look at sort of these molecular explanations as the sort of ultimate explanation, you'll miss a lot of products of cells interacting, of things happening at the cellular level. You'll completely miss those, and they won't be included in your model, and then your model can't predict certain things. Because the spatiotemporal distributions of instructive signaling ions and molecules depend on the local electric potentials, different multicellular potentials correlate with distinct downstream gene expression patterns. So again, these multicellular potentials, which you get at the cellular level, actually correlate with gene expression patterns at the genomic level, at the molecular level. So we know that this is actually quite useful and actually explains a lot of things happening at the molecular level, and we don't have to worry about detailed modeling of that level because our model at the cellular level is capturing it. A central cell, which is the cell maybe of interest that we have, and then there are cells that are neighbors that interact with it, which I called the focal cell, but they call the central cell, is able to measure the number of neighboring cells and learn their bioelectrical state from the downward induced membrane potential changes, which suggests that multi-scale bioelectricity can operate as a top-down mechanism in development and regeneration. This is kind of like the same approach. Well, it's actually kind of an approach you might use, or might be analogous to the whole model of morphogenesis or chemical morphogenesis. So with positional information, remember I said that was a way to describe morphogenesis, to describe pattern formations in cells. And one of the things cells need to know is their position. And the traditional Wolpert argument is that the genome of a cell is the source of that information, that it basically knows which Hough cluster it's a part of. It expresses genes in a certain way to create a gradient or to be a part of a gradient. And then that information is measured by sort of a difference from other cells neighboring to it. So that's how that explanation goes. In this case, what they're saying is that the cell itself can measure the neighboring cells, learn bioelectric states, and they do this not from a molecular mechanism, but from comparing membrane potentials. And so that gives you a mechanism that's very similar to that at a higher level of analysis. Now the question is, maybe there are other mechanisms that we're not, looking at here, and that's fair. But again, we have this model that kind of makes some assumptions about the molecular milieu, and there are a lot of papers on that, so we kind of know those mechanisms that are worked out. We can then focus on the next or higher levels of complexity, higher levels of organization, and then we can merely get correlations with those molecular states, and we can have a predictive model from that. So they talk in this paper about how in biological phenomena, causation is commonly analyzed from the parts to the whole. So that means that in our bioelectric world here, molecular biology is sort of, is composed to produce these cellular states. So it depends on what the molecular state is, and we just sum those together, and we can determine the cellular state. But of course, the problem with that is there are things happening at the cellular state that are independent of that, as well as kind of constraining what happens in the molecular world or the molecular states. So there's this interaction that we can't account for if we just focus on one level of analysis. So they start talking about top-down or downward causation, which is where you have this thing happening at the, say, cellular level that's affecting the molecular level. And if you have interactions between cells, the states of other cells can indirectly affect what's happening at the molecular level of a focal cell, say. Okay, so they kind of get into this. Here we have considered a simple model for the interplay between bioelectricity and transcription in non-excitable cells that explores a top-down perspective. To this end, we invoke an experimentally based model of bioelectrical adaptive transcription that involves feedbacks in multiple size scales, emphasizing the central biological role of the cell. Okay, and so they're kind of getting at this idea of multicellular patterns and biofields, as they call it. I guess they mean like bioelectric fields, but they kind of shorthand biofields. And this sort of approach can benefit from descriptions at scales higher than the underlying molecular events, because top-down control offers new opportunities for external actions. In particularly, multi-scale bioelectricity constitutes a high-level spatial scale. So we have this aspect of the spatial scale between cells across an array of cells, and of course, that array of cells is heterogeneous. So some places in space are different from other places in space. Maybe that's because it's part of a different tissue. Maybe it's because there are constraints on different places in the tissue. And again, like as Susan points out, there's always a mechanics to worry about here. And so this will at least give us some insight into that variation. So multi-scale bioelectricity constitutes a high-level spatial scale control node that expands from the subcellular membrane potentials of organelles, such as lysosomes and mitochondria, to the spatial temporal patterns of multi-cell potentials characteristic of development and regeneration. So this is where we're going from things going on at the organelles, to the cells, to neighboring cells, to ultimately tissues and then organisms. And this allows us to do these kind of large-scale things like development and regeneration. So an organism relies on these things. Michael Levin has done a lot of work in flatworms and looking at regeneration in flatworms. And flatworm regeneration is interesting because it's totipotent, meaning you can take a single cell from a flatworm and you can plate it in a new environment, and it can grow an entirely new flatworm from that one cell. And that means that's because each of the flatworm cells is totipotent, meaning that one cell can produce an entire organism or reproduce an entire organism. And so the thinking of that whole line of work is that this is driven by bioelectricity, that as this one cell reproliferates, there's a bioelectric field that forms, and that bioelectric field is information about what the pattern should look like. So this is like this pre-patterning idea that they stuck into the Wolpert model, where they had to kind of come up with a stopgap explanation for why you have these larger scale patterns. In this case, it's more detailed. You have these bioelectric fields that get coordinated, and so now you have to find a mechanism for that, and that's what they're doing here. We consider here the coupling between bioelectricity and the transcription of two model proteins. To this end, we operationally describe the internal cell representation of the external environment, including the neighboring cells, using voltage-gated ion channels and intracellular junctions. This bioelectrically centered model is complementary to the traditional descriptions focused on biochemical networks and biomechanical fields. Okay, so this is when they get into their model. They get into the membrane potential as their crucial parameter that defines the electric potential difference between the cell inside and the external microenvironment. So you have these easy to define parameters that you can use in your model. They're already used in the experimental models, so they're just used in the computational physical model as well. And so they talk about sort of how membrane potential works to polarized and depolarized cells. While crucial information of the body plan resides within the DNA as a basic single-cell language for making proteins, multicellular potentials can transduce short-term bioelectrical information into long-term transcriptional processes because of the coupling of the signaling ions and molecular concentrations with the local electrical potentials. So in this way, the spatial temporal patterns of cell potentials can provide downstream biochemical information for the establishment of different morphological regions. So you see that there is this voltage or this membrane potential, V, that is really giving you an indicator of the cellular state. And this V is connected then to the molecular world in that it's descriptive. It's a shorthand for a lot of protein synthesis processes, other types of transcriptional processes. We don't need to be too precise in those because all we're really interested in is how V fluctuates and how they contribute to V, the value. And we can look across cells and see membrane potentials for those cells. And then we can ask questions like what happens if membrane potentials synchronize or desynchronize, or if there's long-term depolarization or long-term polarization, or things like that. And then we can go back and infer that there's certain molecular states that cause this in single cells. And so this is all kind of reminiscent, actually, of differentiation waves and some of the mechanisms there that have been proposed by Dick and his co-authors. So yeah, this is definitely that same type of model. And so this kind of goes through a little bit more about their model. This just shows an example of, where you have, say, a depolarized cell being part of a polarized neighborhood, and it has an effect on that neighborhood or on that cell. So this is the same type of thing we think about in cellular automata, where if you have, say, a cell of a certain state and you embed it in other neighboring cells, that it affects its state. So in this case, in A, a depolarized cell is embedded in a polarized neighborhood. So all these cells are polarized with respect to their V value. And in this case, this cell is depolarized with respect to its V value. And then if you put it into this neighborhood, it shifts the behavior of the focal cell to a polarized state. The same thing happens in a depolarized neighborhood. If you put a polarized cell into a depolarized neighborhood, that polarized cell will become depolarized just by virtue of being surrounded by those types of cells. And this is very similar to the types of things you see when you take, say, like a stem cell or a precursor cell, and you embed it into a bunch of differentiated cells. Like if you take a cardiac stem cell and you embed it in cardiac muscle, that cell, theoretically at least, becomes cardiac muscle because it's surrounded by those signals, and then it takes on that state, or it differentiates into that state. So then they just propose a model here where they have genes, proteins, those things through transcription and translation contribute to voltage-gated channels, which are part of the cells that define cellular state. There's a cellular potential regulation or transcription, so the potential itself, if it's changing in the way that we saw in A and B, it can control sort of the rate of transcription for certain proteins. And then the cellular aggregate has this community effect in controlling the states of single cells, and cells themselves communicate through gap junctions or electrical connections between them that regulate the polarization state of the cell. So these cells not only exist as neighbors, but they actually are communicating with each other and sharing information about their state. So Susan says, I found the paper about magnetic fields and artificial microgravity. Moving electrical charges produces magnetic fields, and this is the paper here. So this is cleavage and survival in Xenopus embryos exposed to an 8 Tesla static magnetic field in a rotating clinostat. And this is in bioelectric dynamics. So thank you for that citation, Susan. So finishing up this paper, we end up with a bioelectrical model. This is the bioelectrical model, and you notice that it's got a lot of mathematical components to it. So we're stating all of these things as mathematical functions. We have a depolarizing channel here. We have a polarizing channel here, and that flux is described by these equations here, depolarization and polarization. And from that, we actually get a differential equation, which we can measure the cell state. So we have this, we're basically getting the difference between depolarizing and polarizing channels over time. We also have other equations that we often use for kinetics. So we have, in this case, the mRNA kinetics of protein A. And then we have protein A kinetics. So we're measuring the mRNA and protein kinetics for a specific species of protein. And this is just, we use these kind of mass balance equations to describe sort of the state that we expect from a certain thing that we think is controlling a higher level process. So we just basically say the kinetics of all mRNA are the same, and we're going to look at those dynamics over time. We assume that the fluctuation affects the cellular state, but we don't care about variation in the mRNA structure or sequence. We don't care about the conformational states of the protein. We just care about the mass kinetics. And so we can do this for protein A and protein B and any other proteins we think are involved in this process. And then we can look at that and sum that up, and it gives us a cellular state. Okay, so I think that's enough with that paper. Any questions about that?
Oh, well, I was just finding information about magnetic fields.
Okay.
Because electric fields and magnetic fields are part of the same thing.
Yeah.
So there's some. The magnetic fields in an MRI tend to cause microtubules to align, like they change the functionality of microtubules, so that can cause problems in a cell.
Yeah. 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 Well, thank you for that. Yeah.
Okay, right. I can get the exact reference I've got on the microtubules if you want.
Yeah,
it's the same. It's the same author that I used in my paper on microgravity.
Oh yeah.
He was experimenting with microtubules.
Yeah. Yeah.
Okay, I can get that. The magnetic field tended to kind of counteract what was happening with the lack of gravity, so that you can kind of balance those two out to get things to function more normally.
Yeah.
In microgravity, if you spin it in a magnetic field.
Right.
And I've got a couple of papers for you, so I'll send them.
Okay.
Send them along, even if it's just a reference for a paper. I'll see if I can find the paper. But anyway, okay.
Sounds good.
All right.
This is so it's super interesting. I've been trying to look at kind of more molecular level modeling.
Yeah.
Just this weekend. One of the how to grow almost anything course topics.
Right.
Is looking at protein design. And so some interesting topics around that, mainly just looking at some overlap between some computational methods that I'm already using in brain imaging that would actually be relevant to this kind of molecular modeling, but this makes me think about the UCL professor Nick Lane.
Oh yeah, yeah.
As someone, as well as the most interesting. I don't know if I can remember the name of this book. I think I brought this up, but the most interesting course and kind of book that I did in my biochem molecular biology program was on metabolic chemistry, and this is kind of Nick Lane in terms of just it's not just metabolism, but this what cells are doing, especially around setting up an electrical gradient across the membrane, as being like such a core, such a fundamental process, and just interesting to see, anyway. I've also, Levin hasn't updated his BETC program.
Oh, right.
Which is like the biophysics, bioelectric, I forget what the acronym stands for, but it's his bioelectric modeling tool.
Right.
So it's like been 10 years since the first paper.
Yeah.
And there was a kind of follow-up in 2018, but it was more about adding they kind of added a GUI, I think, but then again, just really another really nice fundamental process to think about and see how it's modeled. See, get into the weeds.
Right.
It's a nice. Anyway, cool to see. Let's see if I can find a spot for it in the. Thinking about trying to do a follow-up supplementary material to the How to Grow Almost Anything course in the fall, where we kind of dig deeper or start to use more kind of production level tools, or and start to explore topics. So this would be a kind of an area which would definitely deserve it. An interesting paper.
Yeah, sounds good. Yeah, thanks for helping us think about that in the context of some of the other modeling going on. All right. So yeah, I think that's it for today. Thank you for attending, and see you next week. All right, bye.
Bye.
Thank you. Bye.